Hello, and welcome to another lesson by GM's Productions. I am Maria Nazarova, and I'm joined today with fellow colleagues Julia, Somia, and Sigourney. Today, we will be discussing the effect of red light on photosynthesis in organic spinach leaves. The purpose of this lab was to determine the effects of different colored lighting on the photosynthetic process of organic spinach leaves. To do this, we used bicarbonate solution in which we dropped leaf chads to see how fast they rose under different colored lamplights. We found that red light sped up the photosynthetic process. Our hypothesis is that red light will speed up the photosynthetic process in organic spinach leaves.
So here we go. This is how we set up the lab. We measure out 1 gram of baking soda, which actually took quite a while for me because I obviously can't measure out baking soda very well. After this, we are going to measure out 100 milliliters of water and mix it in with the baking soda in order to create an environment with sufficient CO2 for our experiment.
We then proceeded to cut out little leaf chads with a hole puncher to create uniform leaves. We will need 10 leaf chads for this experiment, as seen here.
Place the leaf chads into the syringe, absorb some bicarbonate solution, and remove the oxygen from the leaf chads, as shown here. Empty the leaf chads back into the bicarbonate solution in the beaker.
Place the beaker under a lamp and note whenever a leaf reaches the surface of the water. You should record your data every minute.
Hey, you guys, I've been wondering something.
Yeah, what is it?
Yeah, what?
If a plant is sad, do the other plants photosympathize with it?
Oh my gosh.
Maria, don't try to be funny. Anyway, as you can see, after 9 minutes, half of the leaf chads have reached the surface of the solution, so we now have our ET50.
We will now perform this experiment again with new leaf chads and a new solution, but this time the light shining on the beaker will be red.
Geez, you guys, I'm kind of hungry. I could use a light snack. Get it? Like light photosynthesis?
No.
Light? No?
No.
Okay,
No.
well then let's move on. It looks like half of the leaf chads have rose to the top of the solution, making our ET50 8 minutes.
As we can see, the red light sped up the photosynthesis process. In both reactions, however, the leaf chads rose rapidly all at once instead of gradually rising one at a time.
In the control experiment, the one leaf chad remained at the top of the beaker from the beginning, but this is an error because the oxygen was probably not taken out of it. At 9 minutes, 4 more leaves rapidly rose to the top.
In the second experiment, no leaf chads rose until the 6th minute when 1 rose, then in the 7th minute, 2 more rose, and in the 8th minute, 3 additional leaf chads rose. Based on these results, we can conclude that photosynthesis occurs most rapidly when exposed to a red light rather than a normal lamplight.
We hope you learned something new today and didn't fall asleep during this. See you next time, and I'll leave it at that.